At this point in the course, you have learned that the membrane acts as an electrical sink. It can separate the inside and outside environments of a neuron, or any cell really, and hold an electrical charge based on the concentration gradients of sodium into the cell, and especially potassium out of the cell, and on the electrical gradient established by the sodium-potassium ATPase inside at negative 70 millivolts relative to the more positive outside. You know that the neuronal membrane is packed with channels that are voltage sensitive or inherently leaky. You know that the potential of a neuron is locally dependent on what channels are open, and that changes in potential can propagate along the membrane in the form of the all-important action potential. That action potential can carry a signal from the dendrites to the axon hillock, where, if there are enough depolarizing events, an action potential is initiated and propagated all the way to the axonal bouton, or terminal. What you don't know yet is why. What is the point of the action potential? Why waste all of that ATP to establish a resting electric potential just to have it disrupted in a directional way and just dead end at the axon terminal? The answer connects what you know about the electrical properties of the neuron to what you want to know about its chemical properties. You see, neurons are not just electrically active. They also must communicate with each other chemically, and that is generally accomplished by synthesis, packaging, and release of neurotransmitters. Neurotransmitters are packaged in vesicles made up of membrane and shipped to a holding area at the end of the terminal bouton. But unless they're released, those neurotransmitters are useless. So how do they get out of the cell? It turns out that the sudden change in electrical potential due to the action potential opens up certain calcium channels and lets that ion into the bouton where it binds to the snare complex and forces the fusion of the neurotransmitter containing vesicles with the membrane, allowing the subsequent release of the chemical factors. The snare complex is a group of proteins on both the inside of the cell membrane and on the outside of vesicles that fuse to each other and, in the presence of calcium, twist to slam the vesicle against the membrane. Like two soap bubbles coming into contact with each other, the vesicle pops open to release neurotransmitter into the synapse, a space between the axon of the first neuron and the dendrites or receptive area of the next neuron. The neurotransmitter in turn binds to a receptor on a postsynaptic cell and initiates a metabolic process or opens a channel that alters the electrical potential of the receiving cell. If the resultant depolarization is strong or frequent enough, the process begins again. It leaves one to wonder, what comes first, the action potential or the neurotransmitter? The chicken or the egg?